recently, but these are some of these organisms. This is 4 samples of actinomycetes that my students in the teaching lab isolated. So we've just taken little samples. I'll take samples of these again, grow them on individual plates. But so these are 4 different types of actinomycete, and you can see 3 of them are quite similar. They have this whitish layer on the surface. That is a very small microscopic layer of spores that they're producing. That's how they spread.
Spores.
Spores. These are sporulating bacteria. You can also see, if I turn it over, this is slightly darker. So this organism is producing a pigment that is soluble, that is leaching out into the agar. And that's very common in these actinomycetes as well. That's some of the secondary metabolites they produce. The other thing you'll notice, which is a shame you can't video it, but they have a very distinctive smell. What does that remind you of?
Chalk?
Chalk?
Yeah.
I think they smell a little bit like rotting leaves or the forest.
Yeah. I smell of the falls.
The forest or the woods or something. So these are actually producing a compound called geosmin. And geosmin is the smell of the woods, of forests, of rotting leaves. People often smell this smell and say, oh, I'm out in the fresh air, or I'm in a national park or something, and it smells
national park.
Foresty, but actually it doesn't smell like trees or the forest. It smells like these bacteria. That's what we're smelling. And this compound is produced by these organisms. We don't really know why, but it's exploited by a lot of other organisms. So there are insects that will follow that smell because it's associated with these bacteria, and these bacteria grow in areas that are very moist. So if insects in an arid region, in a dry part of the world, want to find water, they follow the smell of these bacteria, and these bacteria grow where there's moisture.
So anyone follows the smells, right?
Yeah.
Of this one.
Yeah, exactly right.
All right.
Yeah. I don't really have very much else going on. This is where my students do their research. Let me see if I have any other plates. These are more. So here's another set of these same bacteria. You can see the very white surface of these. Those are producing spores.
The white one. What is that? I mean, like.
We call them aromycelia.
Aromycelia.
They're very fine projections of growth that have spores on them.
All right.
But I don't have much else going on at the moment, I'm afraid. But yeah, this is my little research space. So we have a microscope and the kind of tools that we need to do basic, characterization of our bacteria. And we have some other tools. This lab is very messy. This is a plan of our research at the moment. And one of my students is actually today is presenting at the undergraduate research conference. We take soil samples from our students, and we take those samples like you just saw, and we build a library of actinomycete species. And then we test in this assay, this growth inhibition assay, we test whether they're able to kill fungal organisms. Then we identify the organism, and then we analyze this data set. We may find that we have a lot of repeats in our actinomycetes library. So we'll refine our library once we know what the species are. And if it's an unknown species, we'll go into characterizing them and maybe collaborating with other chemists and developing those, or pharmaceutical companies. And if it's a known species, we can mine the genome that's already published and identify lead compounds, and again, that would be handing off to another researcher to develop the drugs.
So it's a process of developing the drug?
It's the first step. Of identifying compounds that could be used as drugs. Yeah.
I once learned about it when I was in South Korea.
Yeah.
All right.
And we have other, I'm trying to think of other. I don't have anything else going on. These are our incubators, and this is a PCR machine for polymerase chain reaction, and this is a real-time PCR machine.
PCR, yeah.
Yeah, these are kind of old, but they do the job. They do still do the work. But
I mean, this kind of question is, it might be sound stupid, but how much is it?
This, when it was new,
Yeah, when it's new.
when it was new, probably $5,000, $8,000, something like that.
Oh.
This is quite old now. You can buy them for about a real-time. So because this does real-time PCR, it's a lot more complicated than this basic thermocycler. So it has detection equipment. Probably still $5,000-$6,000 at least. The price has come down a little bit. But,
So another some kind of stupid question. What is the most expensive one in this lab?
that's probably the most expensive piece of equipment. A lot of this is old equipment. You can see this lab is very full of junk because this is all kind of older stuff. But that probably is the most expensive piece of equipment.
PCR.
Yeah, probably.
What is this?
This is an old piece of equipment. I don't know. This is someone who used to work here, who's left, and it is for a microfluidics thing, I think. It has a laser in there. I've never used it. It's not mine. So we share a lot of lab space. So this is very old. I have no idea what it does. But it measures. If I had to guess, it's a small scale microarray, maybe. You can see how old the computer is that goes with it.
Yeah, it's old.
So it's very, very old.
It's old. It looks older than my laptop. Yeah.
Yeah, which is saying something.
Yeah.
Yeah. No, I have no idea what that is. There's quite a lot of old equipment that I haven't used that is not mine. Just like all of this. None of this is my equipment.
What kind of cable is it? I mean.
Oh, I know. Very old. Very, very old. Yeah, I don't think that works anymore. And you can see this is a microarray machine. Now this was probably $10,000 or $12,000 in its time, but this is 20 or more years old. This is the instruction manual for it.
What is this for?
So this was for a microarray.
Microarray.
Yeah, which is a technology we don't really use anymore. But there's all sorts of old junk. I mean, there's a very old stereo, so there's lots of old bits and pieces.
What was the microarray? Yeah.
It was you would input small pieces of DNA onto a chip, and so you could have hundreds and hundreds of samples of DNA, and then pass a solution over, and it would hybridize. It would bind to that, and then light up. So you could. It was sort of similar to. It was a precursor to next generation sequencing. So it was a way to very quickly screen a lot of DNA samples for a specific sequence. So we have better techniques than that now.
So nowadays, what kind of technique are mostly used to screening? Yeah.
Screening. For screening for DNA samples? You mean?
Yeah.
So it's next generation sequencing, which is,
NGS.
Yeah.
Ah.
So that sort of has replaced microarrays. Really, yeah. Yeah. So yeah, it's a very small lab. We can show you some of the research labs further down the hall if you want. There's more space if you wanted to go and look at another lab.
Oh, sure.
Yeah. What time is it? Let's see. I have to make sure.